Okay, here we go again. And in our last video, we talked about DNA. We talked about how DNA is basically providing the code for how we make all the proteins in the body that we need. Okay, and those codes are written in certain areas on our DNA called genes. So that gene expression is basically in what we can also consider is the growth of a protein, how a protein is made. And we've discussed that once at the end of our last video, but it's probably worthwhile going over this again and maybe again and possibly even a third or fourth time so you'll understand how this happens because I think it's really an important factor. Let's go ahead and start this again. We talked about this before, that in making a protein from the code that's on the gene, on the chromosome, there are two main stages. The first stage is what's called transcription. And transcription, what we find is that gene area on the chromosome is exposed. And what happens is when it's exposed, as it breaks down and splits. Remember we talked about the DNA as a two-stranded sugar. They're bounded by nucleotides in between. Okay, bound by hydrogen bonds that actually hold those two nucleotides together. Okay, so what happens is something will break that bond, so allows those two strands of DNA to split. That exposes all those nucleic acids. We talked about that before, the nucleic acids. We talked about those nucleic acids being adenine, thymine, cytosine, and guanine. Now they're exposed. What happens is then we make another single-stranded material from those exposed bases, okay, where we know that thymine will bind to adenine, adenine will bind to the thymine, guanine binds to cytosine, and cytosine binds to guanine. Okay, so as a result, now we make a whole single-strand material with a complementary or the opposite base, and that's what we call messenger RNA. So that messenger RNA has now read the code off of DNA. Okay, it has the code. It's going to take it. Where's it going to take it to? The ribosome. Why the ribosome? Because we talked about this a couple exercises ago. The ribosome is the area where proteins are made. Okay, ribosomes cover the inside of the endoplasmic reticulum, that rough endoplasmic reticulum, and some of the ribosomes are in the mitochondria. Okay, for protein production in the mitochondria, they're used in energy production. What happens is now that messenger RNA leaves the nucleus and then goes to the cytoplasm, to the area of the ribosome. It attaches to R, RNA, and R stands for, of course, ribosome, ribosomal RNA. And the ribosomal RNA, with the code from messenger RNA, is going to send out a message to something called tRNA, and tRNA is transfer RNA that's going to bring the appropriate amino acid that's written by the code. Now, that code from we talked about in a prior lecture is called a codon. And a codon is three of these nitrogenous bases. So it could be an AU, because in RNA, T is substituted for the U or uracil. So it could be an AU, a G, or it could be ACG, or whatever the case would be. And each of these codons is specific for a particular amino acid. So what happens now is that codon that has a specific code, one of these, three of those areas, or three of those nitrogenous bases, that's specific for a particular amino acid, is going to draw a transfer RNA with an anticodon. So if this was the, let's say this one over here is the codon that's on messenger RNA. What's going to happen? Since there's no thymine, it's going to draw uracil, it's going to draw guanine, it's going to draw guanine. And what happens is uracil, guanine would be respective of a specific amino acid. The amino acids that are put in a long chain, and that long chain of amino acids are bound together with bonds, and those bonds then make a long string, which is called a polypeptide, and polypeptides are protein. Another name for a protein. So we start out with transcription, where we transcribe the code into messenger RNA, and then translation is that second process where that code is translated from messenger RNA to transfer RNA to bring the appropriate amino acids. Okay, we'll go over that again. The transcription, again, in the gene, on the chromosome in DNA, is a certain area that codes for a specific protein. And what happens is that code is obtained by that DNA strand, double strand splitting, leaves an exposed base out here, and then what happens is we build a single strand material right here, which is going to be my my messenger RNA with the codon. What happens is that messenger RNA leaves the nucleus, it goes to the ribosome, and the messenger RNA directs the creation of a polypeptide, which is then called translation. Okay, so we talked about that before. And again, the transfer RNA brings the appropriate amino acid. So again, repetition is the way we learn things. Transcription, the process where one nucleic acid, DNA, is copied to another nucleic acid, messenger RNA, or at least that code. An area of the gene on the DNA unwinds and exposes those nitrogenous bases. And on DNA, adenine, thymine, cytosine, guanine. That produces a one-strand, single-strand strand of material that will be able to be transcribed. That transcription occurs when the RNA nucleotides, which is now adenine, uracil, cytosine, and guanine, will be able to form. Now, adenine always binds to where it would be if there's thymine on the DNA, adenine is going to bind to it. If there's adenine on the DNA, it's going to be uracil, because again, in messenger RNA or in RNA, there is no thymine. Uracil is that base. Cytosine is always going to bind to guanine, and guanine is always going to bind to cytosine. Okay. What happens is once they have those complementary bases, then what happens? A sugar backbone is formed. Okay. That creates a single strand sugar, and that becomes messenger RNA. The messenger RNA, which now has the code or the codon, which is 3 bases together, okay, is released and goes to the cytoplasm for making a protein. Okay, that's where the protein is going to be made. Once we get into that second step, after transcription, we end up with translation. Translation, again, is where the nucleic acid RNA is translated into an amino acid sequence or polypeptide or a protein. Same thing, all amino acids. Proteins are polypeptides, which are strings of amino acids. Okay, so the messenger RNA in translation goes to the ribosome, because we know ribosome is the area of protein synthesis. The codons of the messenger RNA in conjunction with our RNA, which is sort of like the bosses. You go here, you go here, you go here. So it directs a sort of thing like that. Attracts the transfer RNA with the corresponding anticodon. So if the codon on messenger RNA was cytosine, guanine, uracil, the anticodon on transfer RNA would be guanine, cytosine, adenine. Okay, we know that. What happens is these will be for specific amino acid. That message nucleotide sequence dictates the arrival order of the transfer RNA molecules. In other words, what they'll do is that the ribosomal RNA will read in series of codons. Read a first codon, brings a transfer RNA. Second codon brings a transfer RNA with the appropriate anticodon. Third codon brings a transfer RNA with the appropriate anticodon. Fourth codon brings the fourth transfer RNA with the appropriate anticodon. So that's what happens. The amino acids from the transfer RNA then form peptide bonds between the amino acids, and they form a polypeptide, which is the protein. It's supposed to be protein, okay? And again, the message nucleotide sequence, or the sequence of those codons, one codon, one amino acid, second codon, another amino acid, third codon, another amino acid, fourth codon, another amino acid, all the way down to the nth codon, which would be the nth amino acid. Okay, and again, these proteins could have the same amino acid but in just different spots. Okay, the result is they have the protein is made from reading the individual codons off the messenger RNA by translating them by bringing the appropriate anticodon from the transfer RNA, which is attached to an amino acid at the opposite end. And those amino acids are linked to create a protein. Okay, so how protein is made, the recipe. Again, we had this before. Let me get rid of me down here so I'm not in the way. Messenger RNA is transcribed from the unzipped DNA in the nucleus of the cell. We know that. Okay. If you don't know that, stop here and think about it. Okay. Pause. Okay, here we go. Ready to go again. What happens is that transcribed messenger RNA, because the code has been read off the code off the DNA, breaks from the DNA, leaves the nucleus, goes to the cytoplasm, to the ribosome with that message or that code. And that code is written in a codon, which is 3 bases in a row. The messenger RNA attaches to the ribosomal RNA in the ribosome, and the message of the code, the codon, is translated. The tRNA, transfer RNA, with the appropriate anticodon to match the codon, brings the correct amino acid. Okay, and the appropriate string of amino acids in the protein sequence is made, hence a protein is born. It's that simple, you know. So it's really not that difficult a process. You only have to remember a couple of bases. You have to remember in DNA, adenine, thymine, cytosine, guanine, and you have to remember in RNA that it's going to be adenine, uracil, cytosine, guanine, and you got to remember that what happens at codon, if I have on my DNA and adenine, thymine, cytosine, what happens? My messenger if that would be a code for a specific amino acid, okay, on the DNA. What would happen is my messenger RNA would be a uracil, because there's no thymine, okay, adenine, guanine. This right here is the codon. But then, when that anticodon from the transfer RNA with the appropriate amino acid comes, the anticodon would be adenine, okay, and there's no thymine, so it's going to be uracil and cytosine. Okay, so as a result, now we basically have the same thing with the exception of the uracil and the thymine. Okay, I'll go over that again. Let's get rid of that. Any 3 that we want. Let's take a cytosine, a thymine, and a guanine. Okay, and let's say that's amino acid Isaac. Amino acid Isaac. Okay, so that's my DNA. Here's my messenger RNA. My messenger RNA is going to read that. It's going to write a guanine. It's going to be an adenine, and it's going to be a cytosine. And that's what we call a codon. That messenger RNA with that codon goes to a ribosome, brings in a transfer RNA. That transfer RNA is going to be cytosine, uracil, because again, excuse me, there's no thymine, and guanine. Okay, so cytosine, uracil, and guanine. That's the anticodon. My transfer RNA keeps on going away. And as a result, that's specific for amino acid Isaac. Okay, that's how we make it. That's transcription, and that's translation. Transcription occurs in the nucleus. The messenger DNA is copied and transcribed on messenger RNA, which now could leave the nucleus. That just shows, here's my DNA, it's unwinding. I make my messenger RNA. My messenger RNA goes, leaves the nucleus, goes to the ribosome. Messenger RNA is transcribed from the DNA message and in a group of codons or a triplet set of bases. Again, there's a triplet, there's a triplet, there's a triplet. Okay? And it's red. And that represents a particular amino acid. The messenger RNA breaks away from the DNA strand. So here's my messenger RNA. It's going to leave. It's going to go to a ribosome. So messenger RNA leaves the nucleus, enters the cytoplasm, and awaits attachment of the ribosome. So now it's going to go to the area of the ribosome with the code from DNA. What happens is, what happens in the cytoplasm at the ribosome is they're going to start to read the codons on messenger RNA and translate them into protein by bringing in the right amino acid in the right order, in the right sequence to be able to make a long string. So the process of translation occurs at the ribosome. Ribosomal RNA attaches to the messenger RNA, okay, which is the site for constructing protons. It's sort of like, if you think about it, if you're riding a bicycle and you have the spokes, or the gear, what happens is the chain is going through that. The little thing sticks up in there and it reads it one after another as the wheel turns, okay? And that's pretty much like what we see happening here at the ribosome. The codons in messenger RNA read by the ribosome, okay? Read by the ribosome. And so now we know what the code is. We know what that code is for that particular protein. The transfer RNA carrying an amino acid with the complementary base pair, or its anticodon, okay, is going to attach to the area of the codon, or read off the area of the codon. And as a result, what happens, it brings the appropriate amino acid for that protein. So here's what we see. Here's the codon, okay? Here's another codon. Here's another codon. Here's another codon, because this is my messenger RNA here. Here's another codon. So if we were looking at this, what would be my anticodon? Well, obviously we'd see cytosine and guanine, uracil and adenine and uracil. So this is my anticodon. This is my code. This is the codon here. The anticodon. So what would it be here? Cytosine, guanine, cytosine would be the anticodon. There we go. Cytosine, guanine is on the way. What would it be here? Cytosine. What? Uracil. Guanine. I don't have a codon here. So here we have a cytosine, uracil, guanine. Here's cytosine, guanine, adenine. And each of those would be for an amino acid. Okay. Now, a couple places here, like this amino acid and this amino acid, they have the same codon, so they have the same anticodon, so it would be the same amino acid, just in a different spot in that long chain. Okay, and that's what happens. That's how we make it. Translation. Also, one of the things we talked about before is when we make that messenger RNA, we have this long string. And the rRNA is sitting there saying, yeah, I don't think that makes any sense here, and that doesn't make it. That's worthless. It's like sometimes when you pick up a book, the first couple pages are blank. What does that tell you about the book? Nothing. Okay, so forget about that. So what happens is actually there is what's called a start codon and a stop codon. The start codon in messenger RNA is adenine, uracil, guanine. So what happens is when our RNA sees adenine, uracil, guanine, it says, "Oh, hey, get ready. We're going now. That's the start. Play ball." That's what happens at that point. At that point, they start to read through all those codons in that order, every three, because the no codon is three. They read that in the messenger RNA, and if for the fun of it, you can actually go through this when you get a chance, and you can read all those individual codons and write down what you think they are. Okay, so obviously A, G, C would be uracil, cytosine, guanine. Okay, everybody knows that. So what happens is there's a start codon, and that starts the process. So once at this point, Yeah, that didn't mean anything. Forget that. That didn't mean anything. Oh, here's that start codon. That means that this protein is going to be here. I'm going to continue on down this road and bringing in the amino acids because now that says start. Adenine, uracil, guanine says start. This is where we start. It's going to read all those codons in a row as it goes down, as it processes that messenger RNA down along the ribosomal RNA. Eventually, it's going to get to what's called a stop protein or stop codon. When it gets that stop codon, it stops making it. That's the end of the protein. So at the beginning of the protein on the code, there's a start. When it's ready, when the protein is made, at the end of the protein, there's a stop codon, and that stops the process. Okay? So translation occurs or stops when there's a stop codon. There are a couple stop codons. Uracil, guanine, adenine, uracil, adenine, adenine, uracil, adenine, guanine. Don't remember these. What happens is I want you to remember that there's a start codon, which is AUG, adenine, uracil, guanine. And there are three stop codons, uracil, adenine, uracil, guanine, adenine, uracil, adenine, adenine, uracil, adenine, guanine. And so what will happen is if this was my messenger RNA, okay, it would start here, and actually AUG, adenine, uracil, guanine, actually is a codon for methionine, which is an amino acid. Okay, here's a second amino acid in the chain. Here's a third amino acid in the chain. Here's a fourth amino in the chain. Here's the fifth amino acid in the chain. Whoops, stop. That's the end. Cut it off right there. Okay, and so we have a start codon and a stop codon. And when our RNA sees that start, it starts to build a protein after that point. When it sees a stop, that's the point it stops, that terminates. Okay, when each amino acid is put together or gets there, it's joined, it's linked by a peptide bond. A peptide bond. A peptide bond is this long chain. If you look at this, this is all these amino acids, a ton of amino acids. And again, proteins are huge molecules. They have lots of amino acids on there, okay? And those amino acids are folded and lumped around and stuff like that. One of the reasons why people can't take insulin by mouth is that insulin is a protein, okay? And what happens is the stomach acid actually takes the insulin molecule, okay, the protein, and unwinds it, unfolds it. And when it unfolds it, it allows it to be destroyed by the stomach acid. So as a result, it can't take insulin by mouth. That's why it's given by injection, because the protein will unfold itself and will become destroyed by the stomach acid. Okay, it breaks those peptide bonds that are between all the amino acids. Okay, so when I have all these amino acids, when the transfer RNA brings the appropriate amino acid, they're linked by a peptide bond, and that forms a protein. Couldn't be much simpler than that. It's a very simple process. Okay, and that's how protein is made. The code's on the DNA. We go from there. And this just shows a very complicated, convoluted picture. Again, here's a cell with a nucleus, and here's a chromosome. We just how it's split. How it happens is here's my DNA going this way. Here's my messenger RNA being made this way. My messenger RNA goes here to the area of the ribosomes, and we start to make a protein off the end of that by bringing in the appropriate amino acid by transfer RNA. That's all. That's how it is. That's how we make these proteins. And just to think of the intricacy of it, but the simplicity of it is really amazing. I mean, just how this came to be is just sort of mind-boggling in my mind. Okay, so let me just show you a little. Oh, here we go. Oh, this is good. Here's the anticodon, the transfer. The transfer RNA bringing it in. Here's another one. They're bringing it in. They keep on bringing these little anticodons to match the codons, and they're making this protein. Here's my protein being made up in there. So there it goes. There it goes. Another one right there. Oh, here it starts again. Okay. Here's another protein or amino acid being brought in there. Another amino acid, and that's how we make a protein. Very simple. Pretty easy. Okay. So what did we go over today? And this is just a little shorter presentation. We talked a lot about this in the last PowerPoint video, but what really happens? I know for a fact there are a number of things that you should know at this point in time. Fact number one, DNA has the code for making all these proteins that the body needs. That's what it's called gene expression. It has the recipe for them all. Number one. That recipe for each individual protein is located on an area on the chromosome, which is called a gene. When I want to make that protein, that DNA will divide, will split, because we know DNA has two strands, has a double-stranded sugar. There's two strands split, which exposes a nitrogen base. Each three of those nitrogen bases on the DNA is a code for a specific amino acid. That code for the amino acid is read by matching an appropriate base, with the exception of uracil for thymine. And what happens is then it creates what's called a codon. For every three bases on the DNA, I'll have three bases on the messenger RNA. That messenger RNA is made in a long strand. That strand leaves the nucleus, gets into the cytoplasm. Where does it go to the cytoplasm? It goes to the site of protein synthesis, and you should know by now that that's the ribosome. That's the ribosome. In the ribosome, the messenger RNA links up with ribosomal RNA, which is made in the nucleolus. That ribosomal RNA says, okay, I'm the boss here. We're going to dictate what's going to happen. It starts to read that messenger RNA in sets of three, or those codons, which is a code for a specific amino acid that the messenger RNA got from DNA. Perfect. What happens is then we start to bring in my son's calling. We start to bring in a transfer RNA with the appropriate anticodon that matches the codon on messenger RNA, and that brings in the appropriate amino acid. And that appropriate amino acid, therefore, then is linked together by peptide bonds to create a long string, which is called a polypeptide. Polypeptides are proteins. So hopefully this all makes sense to you, and everything will fall into place. If you have any questions about this, obviously you could always call me or email me or whatever the case would be, and we could figure this out. But I think we've gone over it now probably about 6 or 7 times, okay? And hopefully after about the 10th or 12th time, it'll be really clear for you, okay? So hopefully everything makes sense, and I'll talk to you next time. Okay, bye-bye.